Good morning, everybody. It's 8 o'clock, so we're going to go ahead and get started. I am delighted to introduce our guest today, our speaker today. I don't think he needs much introduction, but I will go ahead for those of you who don't know him well. I'm delighted to introduce Dr. Lorne Tyrrell, who we're honored to have here. He's a distinguished university professor at the University of Alberta. His research has focused since 1986 on viral hepatitis. He worked on the development of antiviral therapy, which was supported by CIHR and Glaxo Canada. And as many of you know, this resulted in the licensing of the first oral antiviral agent to treat chronic hepatitis B, lamivudine, in 1998. This is now licensed in over 200 countries worldwide for the treatment of hepatitis B. He worked with doctors Norm Kniedemann and David Mercer to establish the first non-primate animal model for hepatitis C virus. A University of Alberta spin-off company in 2003 held an NIH contract for over 15 years to test antivirals for HCV in the only non-primate animal model. Eventually, this company was purchased and remains in Edmonton as Phoenix Bio in 2017. Dr. Tyrrell, as you all probably know, was the Dean of our Faculty of Medicine and Dentistry from 1994 to 2004. And since leaving the deanship, Dr. Tyrrell has taken on a number of important board positions in healthcare in Alberta and Canada. These include chair of the Gardner Foundation Board and member of the Research Advisory Council for the Canadian Institute for Advanced Research. He also has been appointed to the Science Advisory Board to Health Canada, and in 2010, he became the founding director of the Li Ka-shing Institute of Virology at the University of Alberta, where our recent Nobel laureate works. So for his studies on viral hepatitis, Dr. Tyrrell has received numerous prestigious awards, including the Gold Medal of the Canadian Liver Foundation, Office of the Order of Canada, Alberta Order of Excellence, and Fellow of the Royal Society of Canada. He was inducted into the Canadian Medical Hall of Fame in April 2011 and was awarded the Killam Prize Health Sciences in May 2015. Since the pandemic, he's been working on antivirals and vaccines for SARS-CoV-2 and is a member of the COVID-19 vaccine task force that advises the federal government on the selection of COVID-19 vaccines based on efficacy, safety, and timely availability for Canada. So without further ado, I will hand it over to Dr. Tyrrell.
Thank you very much. It's a pleasure to be here this morning and to speak to you about the securing COVID-19 vaccines for Canada, some of the work we've been doing on the vaccine task force. And it's a special day because today is the exact 1 year since the first case of COVID was reported in the United States. It is also the anniversary of when the sequence of the COVID-19 virus was put into GeneBank. So the objective of this talk is to talk briefly about the origins of the SARS-CoV-2 and start of the pandemic. I wanted to go through the methods for producing vaccines because we've really come up with very new and novel ways to describe the safety of the new vaccines and to discuss and determine if the rapid reviews that we're taking, and are they taking any shortcuts? People are concerned, they're reviewing things very quickly, and to outline the concerns over the emerging virus mutants and how the vaccines play into that. So William Olser said, humanity has but three great enemies: fever, famine, and war. And of these, by far the greatest and by far the most terrible is fever. And I think we're living through the proof of Osler's very accurate predictions and assessment. I wanted to show you this slide because it's in 2018, Johns Hopkins put out a warning about pandemics. And it's really remarkable. It said the next pandemic will likely be efficiently transferred from human to human. It'll be non-trivial fatality rate. It would be no widely available medical countermeasures. Immunologically, it would be in a naive population. the capability of the immune system to be evaded by the virus, respiratory mode of transmission, and transmissibility during incubation period when people are asymptomatic, and some percentage of mild infections that people will be asymptomatic. By the way, the current asymptomatic cases and careful assessments in a number of countries is in the range of 17%, and they do transmit, even though they never get symptoms, about 42% as active as somebody who's got symptoms. So that was an extremely accurate prediction of what we're seeing today. The World Health Organization says they are, in 2019, outlined the 10 global threats. And in red are those related to infectious disease, one way or the other. But clearly, the threat of a global, and at that time they thought influenza pandemic was high, and we still think that is high, but it turns out to be coronavirus is the one that is really causing the issues. So the world has had a number of pandemics. And just to put it in perspective, the bubonic plague in the 1340 had world population was estimated to be 475 million, and over 200 million died. So it took out 42% of the population on Earth at that time. Smallpox, 461 million, and 56 million died, and this represented 12% of the world's population. And you can see as we go down, but the Spanish flu in 1918, 1919, 45 million people died, or 2.5% of the population. With the COVID, at the present time, we have a population of about 7.8 billion, and we've lost 2 million people. It's interesting, the London School of Tropical Medicine has estimated that if we had the same type of healthcare system and public health today as we did when the Spanish flu, this number would be closer to 230 million people would have died from COVID. So you can see the public health, medicine, everybody is working so hard, but they're doing a good job at keeping these numbers even where they are. What about these coronaviruses? Well, the coronaviruses were actually not known until 1965. And David Tyrrell, who's namesake but not related, was running the Common Cold Unit in the UK. And they isolated every virus that caused the common cold. And in this, they found some unusual viruses that had these spikes on them and got the name of a crown and jewels on a crown, a coronavirus. And that was in 1967, they were first visualized. By the way, June Almeida was a postdoc from the University of Toronto working with David Tyrrell in that unit. So we have a number of coronaviruses that do infect the human population, and they have probably come in over the years, but then some are mild, but three key ones, SARS, MERS, and SARS-CoV-2 are the critical ones. Just showing the COVID flu has an incubation period 1 to 4 days, an R value of 1.3, fatality rate of about 0.1%. COVID-19 has an R value that now with the new mutants is going up a bit higher, and as a fatality rate of in a range of 2.2%. I would say that there are more asymptomatic, so we don't know if it is that high really or closer to 1%. But SARS had a mortality rate of about 10%, and MERS about 34%. Thank God we don't have mortality rates like that for the COVID-19. So where all of these coronaviruses have come from, really, the big ones, are from bats. And how the bat virus jumps to human is still not entirely known, but there are some, obviously, some concerns with the food chain that might have resulted in some unusual eating habits. But bats that are, particularly this horseshoe bat is one that carries the virus. And we've seen exactly almost the same viruses in the bats as we see in the humans. This just shows you that in two different bats in China, there's high sequence similarities, particularly in this bat, the second one that is in China and Taiwan and some in Japan, where we've seen very similar viruses in bats to the coronaviruses that we see in humans today. So for the COVID-19, there are some critical timelines, and all of this within a year. The first case was December 8th, 2019. It was reported as an outbreak with 27 cases of pneumonia on December 31st. January 9th, the virus was first identified as the cause of this pneumonia. And I would say that January 10th, the sequence was known. It was filed in GeneBank on January 24th. But some of the vaccine companies got the sequence of this virus as early as January 10th, 11th, or 12th, directly from the scientists. In February, it was named the SARS-CoV-2, and the same date, the disease was called by the World Health Organization COVID-19. It was declared a pandemic on March the 11th, and vaccine phase 3 results were coming out in October, November, and Health Canada licensed BioNTech vaccine in December of 2020 and Moderna in December 23rd, 2020. It is remarkable that all of this has happened within 1 year. And to have a vaccine at this stage within less than a year or within the year is truly remarkable. Thanks. So as of January 19th, we have World Health just shows there's 93 million cases or 94 and about 2 million deaths. In Alberta, 117,000 cases with about 1,450 cases, people who have died. Mortality rate a little lower in Alberta. That may just be a reflection of our very good testing for the virus and screening and picking up more cases that were milder, and so the death rate is somewhat lower. So public health measures is the first thing that we do, and we know that everybody knows about this, and I won't talk about it. I just want to simply say that when we began the work on the vaccines, there were about 135 vaccines in preclinical and about 30 in clinical trials. Today, there are about 237 in preclinical studies and 64 in clinical trials. I just want to say the lockdown and the public health measures, the best examples are coming out of Iceland and New Zealand. And surprisingly, there's some communities in northern Italy where we have seen the severe lockdowns has resulted in much less infection. In Iceland, it's a population of 368,000 people, and all of the viruses are screened, they're sequenced, and very careful tracing. But the lockdowns in Iceland decreased the incidence of this disease down to by 98%. So the true public health measures, if you can really have them applied, will have a tremendous effect on this epidemic. On June 6th of 2020, a number of us were asked to serve on the vaccine task force for COVID-19 by the federal government. And the names are here. I won't go through the names, but other than to highlight Monica Nemers as Canada's chief scientist, was with us and has been extremely good. But it's a good committee, and I've really enjoyed working on it. It's chaired by Joanne Langley from Dalhousie and Mark Lenavin formerly from industry. We reported directly to the Deputy Ministers of Health and the Stephen Lucas and the Deputy Minister of Innovation, Science and Economic Development, Simon Kennedy. And our task was to find which of the vaccines were coming out and to find and recruit, to try to help give advice on which ones Canada should procure for a safe, effective, and timely access. Very important that these are the three main things. There is a fourth, and that's how we give some advice on building up biomanufacturing in the country of Canada, because that is an area that we are short on. The Canadian government's role is it completes the procurement agreements with the manufacturers for the vaccines. And then the vaccines must pass through health protection standards before they can be licensed in Canada for use. And then their responsibility is given to the Public Health Agency of Canada, which distributes the vaccines to the provinces and the territories. And as you know, the provinces and territories do carry out the vaccinations and the rollout strategies, how the vaccines will be used in their provinces. Now, some countries have heard of emergency use authorization, particularly in the US and the EU. Canada does not have an emergency use authorization process in Health Canada, but on September the 16th this year, Canada introduced what they called an interim order. And this is in place for 1 year, but I've just heard that it's been extended longer than that. And it was really to support access to COVID-19 drugs and vaccines. So the interim order means that it allows for rolling submissions of data into the health protection branch. Normally, you have to have all the data together and you put it in as a package and it comes out. To try to get more rapid approval, allows for submission of data by the companies on a rolling basis, and it allows to really there is a lot of interaction between Health Canada and the licensing for drugs and vaccines in Canada with other foreign approvals. So they are in constant with the UK, they're in contact with the FDA and the European standards almost on a daily basis on different evaluation. So we do get an expedited review, and it does not involve shortcuts. I just want to, people are worried that we're doing shortcuts with the vaccines approvals. That's not happening. It often improves the thoroughness of the reviews when you involve the other countries and allow for foreign approvals to be used also in Canada so that we should get vaccines approved more rapidly. Just want to emphasize that vaccines are for large populations of healthy people, very different to antivirals, which are directed at people with disease. And you have to have a very, very high standard of safety for vaccines because you're dealing with health people, and you have more ability to take see some side effects in people who are getting the drugs for their disease. So the vaccines, of course, this virus is an RNA virus. It's got a lipid envelope, and it's got spiked glycoproteins on the surface. And the spike glycoprotein binds to the angiotensin conversion enzyme 2, which is the receptor on host cells. And really, you're designing antibodies that will interact with the spike protein and prevent this interaction through our vaccine process. The spikes on this protein are shown in red, and every vaccine we're looking at is looking at how we can get antibodies to the spike protein. Spike protein is this is the genome of the virus. It's a large genome. I would just say that this is a 30,000 base pair genome. Influenza is around 10,000. Not many RNA viruses are as big as this virus. And it does code for 29 different proteins. And some of the structural proteins is the spike protein on the surface. It also has an enzyme, which is an endonuclease, which does some proofreading to make sure that the virus doesn't make a lot of mistakes. And although we're seeing mutations, it's because the virus has some proofreading that we have less mutations than might be expected for an RNA virus. And it also means that a virus of this size can replicate, even though it's an RNA virus. So vaccines educate the immune system to recognize the disease-causing part. The vaccines traditionally have been killed or weakened viruses or purified proteins from the virus. And the traditional vaccine development often takes a long time to go through all the safety and all the testing. Anywhere from 5, 10, or 15 years in the development. So for this COVID vaccine, we've seen the development of some much different and faster ways of getting vaccines. I would point out that this lists all the different ways that vaccines can be produced, and traditionally we've used live attenuated vaccines or inactivated vaccines or protein subunits, but we've introduced vectored vaccines, which is the adenovirus, messenger RNA vaccines, self-amplicating RNA vaccines, virus-like particles, and DNA vaccines have all been in the pipeline for this disease. The one that has really, and I got to say that at the time we were doing this, we were very interested in the RNA viruses, but we're not aware of really how good they have turned out to be. But messenger RNA is, this is not a new approach. People have been putting messenger RNA into people or into animals to try to induce specific antibodies or immune responses to specific proteins. But it's only recently that this has become feasible. The safety, the RNA viruses usually produce the protein within 24 to 48 hours in the host. The messenger RNA does not integrate into host genome, and it's degraded once the protein is made. Most of the RNA of these RNA viruses has disappeared by 5 to 7 days. Their production certainly can be produced much more rapidly. In fact, I just want to say that Moderna got the sequence of the virus, and 42 days later, they were into studies with this virus, with this vaccine. So that is a remarkable short time. Efficacy, the vaccines generate a reliable immune response and seems to be well tolerated in healthy people, and relatively few side effects. Some pain, of course, at the site of injection, a little bit of swelling, but it's no serious side effects. However, as they've gone into bigger trials, we have seen the anaphylaxis in about 1 in 100,000 patients. And the anaphylaxis may be related to some of the components in the vaccine, such as the polyethylene glycol or some of the lipids that are used to get this vaccine into cells. I just want to say that the biotech Pfizer vaccine was developed by a German company, BioNTech, founded by two scientists. They're a husband and wife team. They came to Germany from Turkey, and they've been at the University of Mainz, and they have established BioNTech, a small company. And BioNTech actually teamed up with Pfizer. They approached Pfizer and said they had what they thought was a good vaccine, and BioNTech has become a very remarkable company. The viral RNA is modified, and what they've done is instead of using uridine in the RNA, they use a modified uridine, methyl, and it's a pseudouridine, 1-methyl pseudouridine. This dampens down the innate immune response, and it increases messenger RNA translation in vivo. Because this stabilizes the messenger RNA and it decreases the innate immune response. It encodes the receptor binding domain of the spike protein, and this is a key part of the neutralizing antibodies. And they've stabilized it, and the vaccine is formulated in lipid nanoparticles, more efficient delivery into cells after an intramuscular injection. That has been a key part to come up with the lipid nanoparticles. So this vaccine is delivered with 2 injections, 3 weeks apart. The phase 3 results show a 95% efficacy after the 2 doses. And here's where some of the controversy comes in. After 1 dose, it's 52% effective 10 days after the first dose, or 14 days after the first dose. However, that efficiency has been reported in different amounts. It could be as high as 80%, but a recent report just this week out of Israel, where they've used only 1 dose to try to protect older adults, ended up with showing only about 30 to 35% protection. So it doesn't produce 1 dose, particularly in the target population, the older population, that does not appear to be as good. This is the virus vaccine that has to be stored at -70, and the company has created its own freezers and tracking systems so that this can help with the distribution. The government of Canada has procured 40 million doses of the Pfizer BioNTech vaccine. This is procurement agreements have been signed, and that's what we'll get. However, it's 4 million by the end of March, and then another 20 million into June. So the 40 million are between now and the end of September. That's why we keep saying that we'll be able to immunize everybody in Canada by the end of September. This just shows in the dark dots are severe cases, and in the vaccinated patients, there was 1 severe case. There were 7 severe cases in the placebo. So not only does it protect, but it also protects against the severe disease. This is the interesting slide that after 1 dose, it shows that you can get up to 82% protection. There was a number of patients, about 304 patients in this study, that only received the 1 dose and didn't get the 2nd dose for whatever reason, out of 21,000. And if you just look at protection of the one dose overall, it's 52%. In this subgroup, at long term, it may be higher, and that's why people are encouraged. Maybe we should be using one dose off label, really use. Vaccine, though, really gives about 95% protection when you're using the vaccine as prescribed with 2 doses, 3 weeks apart of the full dose. And the full dose is, thank you. So Moderna is the other RNA virus, this new technology. 2 injections, 4 weeks apart. It turns out to be 94% effective. The vaccine also protects from severe disease, like we've seen with the Pfizer vaccine. The placebo group had 9 patients with very severe disease, and the vaccine group had 0. It can be stored easier, and it keeps for months in a refrigerated temperature, and the cold chain is much, much easier. Canada has also procured 40 million doses of the Moderna vaccine for, again, delivery very similar to what you're seeing with the Pfizer vaccine. Both of these have the lipid nanoparticles encapsulating this modified RNA, and that's the basis of the vaccine. It encodes the S protein of the virus and in special conformation of it, and it makes this is the target of the vaccine. I want to mention that there are co-founders that are Canadians of Moderna. The CEO is Noubar Afeyan, and he was from Lebanon, came to Montreal as a young child, graduate of McGill, and then went on to into MIT, and but was certainly here in Canada for a considerable part of his education. The other one is Derek Rossi, a University of Toronto PhD student, actually of Alan Bernstein, who also went in to MIT, and both of them have been involved in developing companies, and they were co-founders of Moderna. Derek Rossi is not with Moderna now; he's with some other work in stem cell. But there are Canadian contributions to these vaccines that are extremely important. Also, probably one of Canada's most outstanding scientists, Nahum Sonenberg from the University of McGill, has probably the world's best knowledge of RNA and RNA translation, transcription, and conversion into proteins. Nahum is an outstanding scientist and has been a major consultant to Moderna in making this vaccine work. And finally, the whole lipid particles that these vaccines are put in came from Canadians, and one of them at the University of Alberta. Some people may remember Teresa Allen. She retired in 2010, Professor Emeritus from U of A, and now affiliated out at UBC with Peter Cullis. And Peter Cullis and Teresa Allen have had major contributions to developing the lipid particles that these vaccines are put into. So Moderna's RNA vaccine shows that with 1 dose and then 2 doses, it produces levels of antibodies, neutralizing antibodies in patients that are higher than you see in convalescent patients. The large circle is the convalescent patients, and the small circles are different doses of the Moderna vaccine. And it's really excellent antibody levels. And this just shows that, again, the protection is 94%, 95%, so they're both in excellent protection rates. So we'll move from there to another form of vaccine, and new technology is the virus-delivered vaccines. And the one that's getting the most attention has been the AstraZeneca Oxford. This is a non-replicating adenovirus that is put the DNA sequence for the coronavirus spike protein is delivered, and they use the chimp viral vector. The reason they use the chimp viral vector, adenovirus, is they wanted to have no antibodies to adenovirus, and many of us have antibodies, but we don't have the chimp antibodies. So they use a chimp adenovirus, non-replicating, to deliver the S protein. the DNA of the S protein. Again, it's 2 injections a month apart. Phase C results are variable, and there's still some controversy of this because they say overall the vaccine protection is around 70%, but there was a real issue with the study execution. When they used 2 full doses of the vaccine, which is 5 times 10 to the 10 adenovirus particles, non-replicating, they got about 62%. But by mistake, in one of their studies, they had given half the dose in the first dose and a full dose in the second, and that turned out to produce almost a 90% protection. So they are repeating the studies, and the overall effect, they say, is 70%, but we think it'll be higher. And we, as you know, this is a vaccine that has been approved in the UK. Canada has procured 20 million doses of the AstraZeneca Oxford and could have options for more. Just want to say that, again, a Canadian connection to this. Sir John Bell is the Regius Professor at Oxford, very much involved with the Jenner Institute, which is the institute that has produced the vaccine. Another Canadian connection where he is around the world talking about the AstraZeneca Oxford vaccine. So adenovirus, another one that is coming out is Janssen, and Janssen will have their phase 3 results, and I hope that's a very well conducted study, and we're expecting them next week. It's a non-replicating adenovirus, similar to what we're seeing for the AstraZeneca Oxford virus. Its efficiency in trials phase is not known. But there is a very important difference here that is designed to produce protective antibodies with 1 injection. Much easier to give 1 injection than to try to schedule and have 2 injections. So let's hope that it gives excellent results. The vaccine storage is much easier, again, with refrigeration. And in the procurement process, Janssen vaccine, Canada has signed up for 38 million doses. I just want to say again, another important Canadian contribution to this was from Frank Graham. These viruses are produced in HEC293 cells, and the non-replicating adenoviruses were first described by Frank Graham. He was a virologist at McMaster University. One of his key students was Mary Hitt, who's here at the University of Alberta in MMI. We move then into protein-based vaccines, and Novavax is 2 injections, 21 days apart. Phase 3 trials have been started in the US, and we hope to see a rapidly developed phase 3 trial and see if the Canadian government, because this was traditional vaccines, has procured 76 million doses of Novavax vaccine. In the phase 1, phase 2 trials, you can see that it produces very high antibodies after the 2 doses, lower levels after the first dose, and again, the levels of antibody in this are higher than we see most of them are higher than we see in convalescent patients. So it should be a good vaccine as well. Sanofi Pasteur is the other one that has a protein-based vaccine. It's designed to be 2 doses, and its first study started human study started in September. Results are expected in a phase 3 in the Q1 of 2021. Easy to store and traditional vaccine that should produce good immunity. 72 million doses Canada has secured through the procurement arrangement. Finally, there's another one that is a viral-like particle vaccine, and this is being produced by Medicago. Medicago is associated with Laval University and GSK using that adjuvant, and it's 2 injections, and phase 2 and 3 studies are in progress, and 76 million doses have been procured from Medicago as well. So in producing vaccines, you go from preclinical studies of a vaccine substance, then the vaccine substance goes into production and GMP and so all of the things and fill and finish to produce a product that you can ship out to public health. And this is where Canada has done well in the preclinical phases. but we're not in a position to produce enough of the product in our home country. From there, the product goes out for distribution. So the biomanufacturing capabilities of large pharma left Canada as we moved into more generic drugs and less expensive drugs, a decision that was taken by Canadians and to try to get less expensive drugs, but it has resulted in many of the major pharmaceutical companies are no longer in Canada. The other thing I wanted to mention is the Operation Warp Speed in the US. The US has invested $18 billion in 7 major pharmaceutical companies to get 300 million doses of the vaccine as quickly as possible. And this, of course, people wonder why Moderna is producing in the US and Michigan and we can't get any vaccine in Canada. These investments were made that saying the first 300 million doses will remain in the United States. And the problems we're having right now is Pfizer is upgrading its facility in Belgium, and this is our source of the vaccine, and that is a problem. It will be over shortly. So overall, you can see that we've actually got agreements for a lot of vaccines, and in fact, Canada's got the largest vaccine portfolio in the world. You can see that Canada and the United States have procured more vaccines per person than any other country in the world. However, it is the supply chain and when we can get them is the critical issue. We are part of COVAX, and COVAX is an organization to try to get vaccines into many countries, third world countries. As you can see, these diseases are global, so we have to try to find a way to help get everybody in the world vaccinated. And there are a number of organizations involved, but they're involved in COVAX, and Canada is a signatory to COVAX. The United States under Trump was not a signatory to it. The EU is, the UK is, and Canada is. Of course, with the National Advisory Committee on Immunization makes recommendations when these vaccines come out, and these are the recommendations with long-term nursing home residents and attendants, frontline healthcare workers, Indigenous communities, and persons over 80. And I think that this is being followed very closely in most of the provinces. The concern is the mutations. From the UK and South African mutants have been seen. This leads to increased transmission rates, and the R values go up for the virus with the mutant. And this will have an effect on the pandemic. And we really are in a race to get people vaccinated before these mutants become easily transmissible and start spreading the disease quicker. So the UK mutant is a B117 in England, first described, and it is 1.4 to 1.8 times more transmissible than the original strains. And this means that it's more difficult to control, and the doubling times will be faster. And there have been models made, and in the UK right now, about 50% more transmissible, and it's samples in 86,000 Brits recently that have got this mutant. The mutant has been spread around the world and is appearing in the United States, Canada, and in these countries. Most of it has been through importation by travelers, but we do have some of this virus growing in our population. The wild virus and the UK virus, just say I won't any details here, but there's 17 mutations in the UK strain. The key is the mutation of N in position 501 to a Y. This is an asparagine to a tyrosine near the top of the spike, and it increases the binding to the ACE2 inhibitor or the ACE2 receptor, and it's only attached by one of the spike protein is a trimer, and simply binding one of these trimers to the ACE and the N501 attaches to this much better than the wild strain. You can see this as the N501. The interaction is not as good, but the tyrosine interacts much better. This just shows you that this binding is tighter than it is in here with the asparagine, and now this is resulting in increased transmission. And on modeling, this is modeling for Ontario. Blue is the current wild strain, and the pink is where we can go with this strain if it becomes easily and more integrated into population. The mutant virus takes over and replicates faster, so it will take over as the predominant strain. Fortunately, the first testings of the vaccines against the mutant strain show that it is neutralized. This strain is neutralized by the BioNTech just as rapidly as we're seeing with the wild strain, or even a little bit better. The other big question is, can we get more people protected using a single dose? And can we have more people protected by just delaying the second dose? There is no experimental evidence that it's safe to deviate from the tested protocols. The second dose increases the levels and the durability of the immune response. And one of the concerns with this one dose is people will assume because they've been vaccinated that they are protected when they're really not. They may not be. And the mutations of the virus are emerging, and partial immunity may help select for viral strains of the virus that are resistant to the vaccine. And in the older target populations, when you're using one dose, it's harder to get an immune response. This might be an ideal situation to have not only mutants that spread easier, but our big concern is, do we get mutants that might be able to evade our current vaccines? Israel's study just come out this week that shows single doses in older people yields about 30% protection. And so at the present time, we don't feel there's any reason to deviate. There's lots of pressure to deviate, but there is no good science evidence that a single dose would be the way to go for the current vaccines that are licensed. Vaccine hesitancy is still a problem. It was in November, we did surveys and it looked like 30 to 40% of the people were hesitant about the new vaccines. This will delay. The reason you take a vaccine is to protect yourself, but also develop community immunity and herd immunity. Anti-vaxxers are always about 5% of the population, and they're very rigid. We don't waste a lot of time trying to convince anti-vaxxers, but vaccine hesitancy are the targets. Many people, yes, they will take the vaccine, and a study just done in Canada last week shows that now Canadians, 80% will take the vaccine, up from about 60% in December. Communicate early and often about the importance of these vaccines. Listen to people's concerns and be responsive. Be honest and frank and transparent about the vaccines. Speak clearly and with compassion, and build trust in science. This is the way that we'll be able to convince that middle group with vaccine hesitancy to take the vaccine. Conclusions, we have very positive results, particularly with the COVID-19 mRNA vaccines. This is a remarkably fast vaccine that's been developed. It is safe and it is effective. Early results show the vaccines prevent severe disease. We hope the vaccines will likely end the pandemic. There has been significant Canadian contributions to COVID vaccines, and rollout times started in December, and Q1 of 21 was to have 6 million doses, 2 million from Moderna and 4 million from Pfizer. We are behind in getting those supplies, as everybody knows, and it's in the paper, likely weeks, but not months behind some other countries in getting the vaccines, and I've given you some of the reasons I think that we are behind. So there are key questions that remain. How long will immunity last? There's more and more studies showing that it will last a significant time. We're up to 8 months now that patients, that there's still evidence of good immunity. Phase 4 studies for safety. Phase 4 studies really revealed some of the problems with the anaphylaxis that is present with these. The vaccines remain effective as the virus mutates so far. Boosters will be required. We don't know, but it's possible. Are the vaccines more effective? Are some vaccines more effective than others? Particularly if we get into mutants, we'll be looking at all the different types of vaccines and how effective they are. And how do the effective vaccines? Well, now we have no placebo groups to compare to, because we should be seeing vaccines compared to the standard of care. And will the viruses, the vaccines reduce transmissibility? This is really related, need more studies on that. It gives protection, but not necessarily transmissibility that we've seen. Thank you very much, and I'll stop there.
Thank you, Lorne. As expected, that was fantastic. Really amazing. There's lots of questions in the question box, and I'll go through most of them. But if we don't get time to answer them, we will ask Dr. Tyrrell if he can provide some additional answers. So there are some questions around, the delay in Alberta that we're all very well aware of, and some worries about second dose. So we know that at least within Alberta, they've increased the interval for the second dose up to 42 days.
Yes.
Do you have any concerns about that, or do you have concerns about the fact that right now we don't have procured doses? Is there going to be risk to Albertans based on that?
Yeah, I would just comment that it's not Alberta targeted, it's Canada targeted, and it's not even Canada targeted, some of them. But in the EU and in the US, they're going to get access to these vaccines sooner than we are, based on their heavy investment in it. I'm not as concerned with the delay between number 1 and number 2 doses up to 42 days. And I think the National Immunization Committee has been easy with that. It's when you get into longer delays, the first vaccine can see waning of the immunity, and particularly in older people, if we try to stretch that out too long, I think that there is a possibility that we get ideal circumstances to see the immersion of viruses that have mutants that may be resistant to the vaccine. So that's a concern that I have. But I do think we can go at least 6 weeks and maybe longer. It's not following the recommendations. We have somebody on our committee that is an ethicist, and she is saying, if you're going to do this, shouldn't you have patients sign consent forms, because you're really experimenting when you start changing the doses and the intervals.
Yeah. Okay. There was a question around the number of doses that Canada has procured. That nice slide that you showed with Canada and US. Do you have some philosophical thoughts about that? And secondly, I guess what's going to happen with those extra doses?
Yeah, Canada has procured more doses than just like the United States is trying to procure a lot of doses, and we have close to 10 doses per person, meaning 2 doses in most of them. So we got 5 times as much if we get it all coming through. There are some conditions on those procurements. They have to meet the standards that they're approved by Health Canada before we're dealing with these. The second thing is that Canada will be donating some of the extra doses through COVAX to developing countries in the world that we really want to see the whole world get vaccinated for this virus to try to stop the pandemic. So I think we're going to see that. The other thing that we don't know is what will happen if we need boosters. And will we use up a lot of those doses in boosters? A lot of them vaccines that we're procuring are the proteins. They're very stable over a period of time, and you could be using them for other boosters if they're needed in the future. The whole idea here was that at the time we started from the vaccine task force, we tried to recommend what we thought would be the most effective vaccines. We started out with 129 candidates, and we looked at a lot of those. There was 40-some meetings that we had that often lasted 2 to 3 hours, and interviewed many of the companies. And from there, we tried to make judgments of what vaccines should Canada try to get. And we did have high on our list both Pfizer, BioNTech, and Moderna, as well as AstraZeneca, and Janssen, and Novavax.
Speaking of vaccine efficacy, one vaccine group you didn't talk about was the Russian vaccine, and I know there was a question around that. What are your thoughts about that one?
Well, some of the Russian vaccines and the Chinese vaccines, there's quite a number of inactivated vaccines. They simply produce the virus and inactivate it. And inactivated vaccines that usually have some more side effects, but our concerns is that some of the studies that have been done in Russia, in the military and other things have not had the standards that we would use for approval of safety of vaccines in Canada. We usually like to look at 34 or 40,000 patients that receive it to show the efficacy, but also to show the safety. And the other thing that is coming out is the inactivated vaccines out of China and out of Russia that have been used in some countries like Brazil and Indonesia have shown much less protection than they originally thought. They're down in the range of 50 to 60% protection rather than 90% that we're seeing with the messenger RNA vaccines.
There's an interesting question in the group that I'm just going to ask. You've talked about mutations, and I think that's a big concern that people have in terms of vaccine efficacy. In your opinion, Lorne, how quickly do you think that companies can change and pivot and develop a new vaccine if there is, significant mutation?
That's a wonderful question and a very important one. I think that we have never used and depended on artificial intelligence more, but I think that we're going to be looking at trying to collect any signs of a vaccine that becomes a mutation that is resistant to the vaccines. This is an international effort to collect such specimens and to rapidly get it into particularly the mRNA vaccines, because there you can start designing the messenger RNA to try to counteract those changes. And as I pointed out in the beginning, from the time that Moderna or Pfizer biotech got the sequence, they were in production of vaccines that were going into trials in 42 days, or you can see how short it is. And that's because of the technology. You can modify the RNA, you can put in some changes, and that can be done very rapidly, much more rapidly than any traditional vaccines. So I think it'll be a battle. I think it's a race to get the world vaccinated and get this disease under control before we see the emergence of some mutants that might be resistant. And if we do see that, this will take international cooperation and our most powerful intellectual abilities to design vaccines that will be able to counter that.
Thank you. I'm going to ask you a little philosophical question that our friend Dr. Paul Armstrong came up with, and that's related to our domestic capabilities to make vaccines. And do you think that this deficiency will now be addressed by your committee, by Canada, and so on? Do you think?
Well, let me just say that we need to address that. And we had plans there was a plan around the HIV vaccines and potential building more facilities in Canada in 2008, 2009. All of those were put on hold. We don't have major companies that are producing vaccines in Canada outside of Glaxo, and Glaxo is making the flu vaccines in their plant. But without the companies here that are doing this, it's been hard, but we need to invest. Canada needs to invest in biologicals, how we produce biologicals in Canada, including vaccines and other biologicals. And we have given the evaluation, we've seen the increased investment in NRC in Montreal, we're seeing increased investments in Vito, you've seen increased investments in Abcelera, which is a very successful company in Vancouver, and we're looking at other potential ways that we can increase the production. The real problem is to increase the production, it takes a bit of time, and it'll take at least 2 years in many of these cases to get vaccines into production in new facilities. But there are investments going into new facilities to try to address that very significant shortage in the country.
I know it's 9 o'clock, but I'm going to ask you one last question where there was a lot of chatter around, and that's, given your role on the vaccine task force, do you have any insights as to our current urgency and the capabilities? Do you have some ideas of how quickly that supply chain is going to pick up? I mean, you talked about the changes that are happening in the factory and so on that's limiting it, but
that I got to say that there was a lot of relief when we saw the results coming out from the mRNA vaccines. for a respiratory infection to get a 95% protection was fantastic. We don't ever see that with influenza vaccines or others in respiratory diseases. So there was some, I would say, some relief to see those numbers. There has been some concerns as the supply chain has not been lived up to what we thought it was going to be doing. So we're very concerned and making sure that we have a plan B. The plan B may be the adenovirus is delivered as a backup and as a if we're going to get Canada vaccinated, every Canadian vaccinated by the end of September, as is promised, we need to be doing 200,000 or more vaccinations per day between now and then. There's two vaccines, and you take people over the age of 16. There's 26 million in Canada. Just put the figures together, 52 million between now and September injections. We need to do 200,000 a day. We're not close to that. We need to get the supply up, and we need to, and it will go up. And we need to see if there's approvals of the others. There will be a choice of vaccines. And if we see that the adenovirus delivered vaccines have something in the range of 80 or 90%, we'll be using those. So it is a matter of getting the supply. I think the province and territories are going to get it out, but you're going to need to do 200,000 a day between now and September.
Quite the logistic nightmare. Thank you so much. That was, as promised, an amazing presentation. We've gotten lots of feedback that it was outstanding. I'm hoping to get you back to do a part 2 later. So thank you so much. We will reach out to you if it's okay to help with some of the questions that weren't completely addressed. But on behalf of the department, thank you for all you're doing and for taking the time to present to us today.
Well, it's my pleasure, and thank you very much for inviting me.
Okay, take care.
Bye-bye.